today we're going to talk about a very important pathway of immunity, and this is the adaptive immunity, and it's equivalently, let's put it that way, it's equivalently as important as innate immunity, and we're going to spend at least a lecture or two on innate immunity. But today we're going to talk about adaptive immunity, and we're going to talk about a very important part of adaptive immunity, namely humoral immunity. So in terms of adaptive immunity, really, adaptive immunity is the immune system's way of creatively configuring and identifying the brand new pathogens it has never seen before. That's one function. And it also has a memory bank. It has a bank of cells that were previously exposed to pathogens, and they've remembered the shape of the pathogen, and they can be called up into action very quickly. So even though adaptive immunity essentially only includes two cells, the T cells and the B cells, in my opinion, it's the more complex arm of the immune system. So really, adaptive immunity is initiated when there is a dendritic cell that engulfs a pathogen. And we have the thymus, right? So here's the thymus, and which is a source of where the T and B cells reside. And we also have the precursor bone marrow. And so the precursor bone marrow releases cells that are lymphocyte progenitors, which means they're pre-lymphocytes and they can become T or B cells. And then we have T and B cells essentially residing in the lymph nodes. And these B and T cells are stimulated to proliferate when we have these viruses and bacteria that present themselves to this adaptive immune arm of the immune system, So, and during the process of adaptive immunity, we both identify the pathogen, we determine that the pathogen is different from the human organism and the various human tissues, and then we differentiate cells that help to facilitate other cells coming, and then we basically eradicate the pathogen, and then we leave some cells behind that will remember the shape of the pathogen in case the human organism experiences future infections from that same pathogen. So humoral immunity is basically the arm of the immune system that involves B cells. And it's one half of the adaptive immune system. And there are various hallmarks that we're going to talk about. We're going to talk about how B cells differentiate, why they differentiate, what their function is. And then we're going to talk about how they go on and execute their eradication of pathogens. And then finally, as we discussed, they actually remember the pathogen. So in terms of development of these immune cells, you can see we have our bone marrow precursor. And then in terms of, we're going to talk in this lecture about the B lymphocytes. And a nice way to remember B lymphocytes is to think of B as representing bone marrow. They also are aligned with the bursa. And they can come to us from several places, but once they get to that precursor stage, basically they reside in the lymph nodes. And that's one of the reasons, for instance, when you have some kind of respiratory or viral or bacterial infection, often your lymph nodes get swollen, and that's because we're having all of these cells get produced, both the T and the B cells. So the B lymphocytes develop into plasma cells, and it's the plasma cells that produce the antibodies, the classic antibodies that are associated with humoral immunity. So the B cell, remember, is produced in the bone marrow. It migrates to the spleen and to the lymph nodes. And the outer surface of the B cell has what's called a B cell receptor. And the B cell receptor is activated by binding to an antigen. So it's almost like an outer band. Just think of it as like an outer band that's sticky and is almost like clay. So when the pathogen comes along and sticks to that outer shell, that B cell configures itself to that shape of that particular antigen. So that's the way I really like to talk about it, that part of the function of these cells is to really determine that indeed a pathogen has invaded the body, and then learn about that pathogen in terms of its shape, so that then other cells can come along and recognize the pathogen and then eradicate it. So we have initial activation of that B cell receptor binding to the antigen, and that activation actually produces antibodies, and these antibodies are specifically configured to fit to this antigen. And then also we get secondary signaling of the T cells, and that's a whole nother process that we'll talk about in a different lecture. So the hallmarks, now this conglomerate here, we couldn't make it any bigger than it is. I'm not sure you would want to have it any bigger, because it shows the complexity of both innate immunity and acquired immunity, but most specifically, it shows the complexity of how these B cells actually interact with essentially all components of the immune system. So the B lymphocyte is the central cell. It responds to these extracellular antigens. It's antibody mediated, it's antigen specific, and most importantly, it's highly complex. So in this course, we've tried to take this complexity and explain it in a way that it becomes very clear and, most importantly, very easy to remember. So in terms of antigen recognition, the B cells in the serum and in the lymph nodes recognize these extracellular antigens. The receptor is highly specific for each antigen type. Remember, it's like a clay, and so when those unique antigens come along, it fits itself to that particular antigenic shape, and then it remembers that shape, and it uses that shape as a template to actually make more cells. So these antigens then are on that B cell receptor, on that pathogen surface. We have various unique shapes. Now remember, the bacteria lectures and the virus lectures and the parasite lectures, all of those organisms, if you can remember, are very, very complicated, number one, and number two, they're very uniquely shaped. So these antibodies are very clever because they find that little piece of that particular organism that is unique to that organism, and that's the place that is remembered. So we have microbial protein structures that can form these antigens. We have lipids. Remember, lipids are fat molecules. And then we have various chemicals. So we have ions, we have different carbon configurations, we have particles, we have all kinds of things that can be remembered. But there are some diseases where they are so variable that basically they can't be remembered. And one of the prime examples of that is HIV. Now, HIV, number one, continually evolves new shapes. And so even if the immune system remembers what it looked like before, it's not going to remember what it looks like now. So that's another challenge that the field of virology and microbiology is certainly facing, and we've discussed that in various other lectures. So here's an example. Now, we know, for instance, that the protein molecule is the one molecule, the one biomolecule that is the most complex in terms of shape. So it would make sense that we would create antibodies to these uniquely shaped proteins that are on bacteria and viruses. But also we can have complex lipid molecules, and those lipids can actually be the place where we remember, because that's the particular part of the pathogen that's unique. And then we have these chemical antigens. Now remember, in the infectious patterns lecture and in the bacterial adaptation lecture, we talked about toxins. Now, toxins are often not bacteria. Remember, they're secreted by bacteria. And these toxins are actually chemicals. They're poisons. They're proteins. They're various kinds of things. So sometimes it's not the bacteria that is remembered, but actually the toxin, because it's the toxin that's actually the damaging molecule. So the clonal expansion, so once the B cell figures out the unique shape of the antigen, it activates and it begins to divide. And the way in which it divides is it divides into new B cells, all of which remember the shape of that antigen that encountered the initial B cell. So that receptor becomes highly specific for each antigen type. And for the particular antigen of the pathogen that's invaded, that's the shape that gets replicated, or that the cell with that shape ends up getting replicated, and it gets replicated and it gets replicated. So then we actually have very many antibodies and very many B cells that are all uniquely configured to that particular antigen shape. So a clone is actually a group of genetically identical naive B cells that have configured themselves to these particular antigens. So here we have a naive B, and then it differentiates, it becomes activated, and then it actually goes on through the developmental process and it becomes a plasma cell. Now, it's the plasma cells that go on to actually make the antibodies, and it also can become a memory B cell, which is the cell I told you that remembers the antigen shape, and after the infection is healed, those B cells stay around. Now, in terms of maturation, this is a different picture of maturation. So we start out with our precursor cells, and we go through various maturation stages. And then here, this part here is actually where we start expanding that clone of B cells to be complementary to the shape of those antigens, or that new antigen that came on the scene to infect the host. And then these antibodies, okay, these antibodies right here, all of these antibodies are creatively and uniquely shaped to also fit that antigen. So now we have all of these floating complementary shapes to the antigen that once they attach to the antigen, basically the antigen, the day of the antigen is over, okay, because then those antibodies call up all these other cells. They all recognize that indeed this pathogen is the one that we're going to take out, and then they eradicate the pathogen. So differentiation is where these cells, the clones, remember, differentiate into plasma cells, and it's the plasma cells that produce the antibodies. So these plasma cells, that this word, by the way, is secrete antibodies that are specific to that originating antigen. And this process, this length of time that it takes to make the antibodies, it's a delay. Sometimes it takes a little while. Now, a good example of that process is, for instance, if one gets infected with HIV, sometimes it takes up to even 6 months before the antibodies appear. So if, for instance, if one thinks that they've been infected by HIV and they right away go and get an AIDS test. And remember, the AIDS tests actually determine whether or not antibodies are in the serum. And if they are, then you can assume, of course, that you've been infected by HIV. Now, sometimes that process takes up to 6 months. So if you go too soon after thinking you were exposed, sometimes the test will be negative. And that's not the time to go out and be happy about it or celebrate. You need to wait a full 6 months and then get tested again. And then if you're negative, then you can pretty much assume that you were not infected with HIV. So this process of taking the time that it needs to actually create this clone of B cells and then have the B cells differentiate can sometimes take that long. Now, these cells end up becoming plasma cells, and it's the plasma cells that secrete the antibodies that, remember, are specific to the original antigen. But we also have these other cells. Now, the B cells can also do what's called presentation. They can present the antigen, and some of the B cells are involved in that process. So they present the shape of the antigen to T cells, and then the T cells respond in their own way, right? They have their own set of weaponry, and they respond with targeted lethal chemicals and lethal cells that eradicate the pathogen, and that's going to represent an entirely separate lecture. Now, elimination of the antigen now can be done several ways. One way is by a process called agglutination, where you get clustering of these antibodies that attach to the antigen, and then that actually marks the antigen for destruction. And these immune cells called macrophages come on the scene, and then they eradicate the actual antigen. And these macrophages also activate the complement system. Now, remember, we had a whole separate lecture on complement. And remember, complement is part of the innate immune system. But, if you remember too, complement has its own independent action on these pathogens. And sometimes pathogens can be eliminated entirely without even making it to the adaptive immune system. So we stop the immune response when the antigen is eliminated, when the antigen-specific cells go through apoptosis, which is another word for cell death. And then we have other plasma cells that end up going back, so to speak, to the bone marrow, and these are the memory B cells, and they remain in the bone marrow and back into this immature state for a variable length of time. So in a sense, basically, we're going back to the beginning. So here we have the antibodies that are involved in destruction of the antigen, and then we mark the antigen for destruction, and then we activate complement, of course. And so this is the whole process by which we eliminate the antigen. Now, finally, this is the part I was telling you about going back to the beginning. We have the component of the immune system has a memory, and this is very, very important. So both B cells have a memory component and T cells have a memory component. So the memory component of B cells is where we have the shape of the antigen that's retained on some of the B cells, and that these B cells go back to the bone marrow where they stay, and they stay with the shape remembered. So both IgG, which is a particular class of antibodies and a particular class of B cells, and IgM is the class of antibodies that are involved in memory. These two classes of antibodies and B cells stay back on the scene, and by remembering the shape of the antigen, they can execute and be called into action in a much quicker length of time than it took in terms of the original infection. Often takes days, weeks, and in the case of HIV, takes up to 6 months, right, to actually have presence of antibodies. So once you've been exposed to a particular pathogen and you have memory B cells, for instance, then the length of time it takes to get activated is much shorter. So sometimes these IgM or these B cells that have memory survive for years, sometimes for life, and sometimes not. Sometimes they, unfortunately, our immune system forgets the exposure, and then you can be exposed again, or you can develop the infection again. So if the memory is active, you can have a much more rapid response. And this principle of memory is what vaccination is completely geared around. Because what we do is we administer vaccines to folks. And in a sense, we are teaching the immune system what the shape is of the pathogen without making the pathogen lethal. And then the immune system develops B cells just like it would for a normal infection, and it develops memory cells, and these memory cells stay around. But we know, for instance, with some vaccines, you need to get a booster because the human host can forget, right? They can forget the infection, or that the memory B cells, for whatever reason, just, over time, just go through apoptosis themselves, and then you don't have any more memory cells, and then, you get a booster vaccine, and then your immune system is taught again what the shape of these antigens are. So here's a picture of what I call going back in time. So we had our antibodies, right, that took out our pathogen that originally infected the host, and then we go back. It's almost like we go backwards in development. So we go back to the plasma cell, we go back to the B cell, and then that B cell goes back to the bone marrow as a memory cell, and that memory cell stays in the bone marrow as long as it can, right? And then if another pathogen comes along, those B cells are primed and already shaped and developed to go. So here's another description of that process. So this is the IgM. Remember, M stands for antibodies. And what the way I like to explain it to students is I like to talk about what happens, for instance, if you get locked out of your apartment. And I don't know if that's ever happened to you, but it's probably happened. Well, it's happened to me. Okay, and so what do? You get on the phone and you call the locksmith, and they come out to your house, and they have a huge keychain of all these different kinds of keys. And so then they try to find the one that they think pretty much fits your lock, and then they go and shape it. Well, that's what I like to tell students about with these IgM antibodies, because this particular antibody really reminds me of a locksmith keys, or locksmith a ring, right, of all the different keys. So here you will see, here's an IgM, and it has all of these different shapes and all of these different places where shapes can be remembered. So it's almost like a locksmith ring with a whole lot of keys. And so with these IgMs, they have to remember all of the infections that we've ever had, right? And so, so the way it looks, right, the way it looks chemically is it has all these places, right, for things to fit in. So basically that's how all these different infections are remembered. So what happens in our first exposure is it takes a period of time for our immune cells to recognize the pathogen and then go through differentiation. Well, the second response, so the second time, right, we're exposed to that pathogen, Boy, so we don't have any lag time here. We are just primed and ready to go. So the presence of antibodies, the IgM and the IgG, get immediately launched. Okay, so now we have some questions. So the first question has to do with what does it mean if you detect antibodies to a pathogen in a patient's blood sample? Well, it means that the patient has been exposed to that disease. Now, if the patient has had a vaccine, right, then those antibodies essentially represent the fact that the patient has had a vaccine. If the patient has not had a vaccine, that those antibodies represent the fact that the patient has actually encountered the pathogen. So so as I've talked already about with the HIV test, that's basically what we're testing for, is we're testing for antibodies to HIV. And if we see that there are antibodies to HIV, we know that the person has been exposed. Now, clearly, we don't yet have a vaccine for HIV, but we have vaccines for quite a lot of other infectious diseases. And, potentially having antibodies can tell us if the person has been exposed to the pathogen or had a vaccine. And sometimes what we do is we draw what's called an antibody titer, where we actually determine whether or not the patient actually needs a second vaccine or needs a booster. So if, for instance, especially with hepatitis B, we can take an antibody titer and we can see, are the levels of antibodies sufficiently high enough for a second infection to be prevented. And if not, then we can give the patient a hepatitis B vaccine booster. Okay, and so with a lot of other diseases, we can do that as well. So the second question has to do with what happens with the antibody level after a long period of time following the original infection. So that question really gets back to this whole memory issue. So what constitutes the length of time that our immune system can remember infections? I mean, I actually think that's a highly variable process. So especially if, for instance, the individual is experiencing immune suppression for some unknown reason, then potentially the antibodies that were there are just so weakened, or just the levels have been so depleted, right, then the memory is essentially gone. And as I've said before, you can actually give booster vaccines. And then the other thing is you can, if you know, for instance, that the particular disease might be active, then you can keep the patient away from being exposed to that disease, particularly if they're immune suppressed. So the last example has to do, or the last question has to do with, can you directly treat patients with antibodies? And how does that work? And give examples. So for instance, say, there's a couple of diseases that come to mind, and they're both serious. One is rabies, okay, and the other one is tetanus. Okay, and rabies, remember, is a virus. It's a viral infection. And the manifestations is a very, very serious disease. So sometimes if an individual suspects, for instance, that they've been bitten by an animal that might be carrying rabies, instead of waiting for the individual's immune system to recognize the rabies virus and then launch its own immune attack, what happens is the patient, even without knowing that they've been infected or not, they're given what's called gamma globulin. And what that is those are actually antibodies that have been bioengineered and created that are directly targeted or directly targeting the rabies virus. So essentially, you're given rabies antibodies up front before your immune system even has the chance to create its own. And the reason for that is because rabies is such a serious disease. Now, the same thing, now tetanus is a bacterial, and it produces these endotoxins, and they're neurologic in function. And what they do is, I mean, basically tetanus, if you get infected by the tetanus bacteria, then you could essentially become completely paralyzed. It's a very devastating disease. So you can actually also get antibodies to the tetanus bacillus or the tetanus bacteria, and then that actually will enable you to potentially prevent the infection from occurring. And I know that happened to me once, where I didn't get rabies antibodies. I actually got tetanus antibodies, but I was bitten by an animal, and I went to the emergency room, and they immediately gave me these tetanus antibodies, and then eventually I got a tetanus vaccine. Now the tetanus vaccine is combined with other vaccines, and so really getting tetanus now in the United States is a very rare occurrence. But it's a very rare occurrence because so many of us have actually been vaccinated. So that concludes our lecture for today. Thank you so much for visiting educator.com.